SP5 will study trophic interactions across tree regeneration stages, and this will be done by the PIs Tim Diekerter and myself, Alexandra Erfmeyer, and it will be mirrored by our Chinese partner project headed by Zheshu Qiao from Beijing. Coming from a plant ecologist perspective, one is primarily interested in understanding plant diversity effects on tree growth and forest community structure, but it is even more comprehensive when focusing on the role of multi-trophic interactions in mediating these relationships. Forest community structure and diversity are largely determined by tree reproduction and seedling establishment. But beyond the seedlings, there are even many more stages in the tree reproductive cycle that are of importance, such as the seed set, dispersal, germination, or even seedling growth. And all of the transitions there in between are largely affected in multi-trophic interactions, as there are pre-dispersal seed predation, post-dispersal seed predation, up to damage and infestation of the seedlings. So there is a multi-stage process of plant recruitment that is susceptible to various factors. First, the local environmental context is crucial for demographic processes, such as seen here for seed deposition or seed predation. Each of these processes may vary independently or differently in response to different environmental variables. Second, we know about the existence of sort of exclusion zones around an adult tree. That means zones within which a tree cannot replace a conspecific dead tree. So at local small scales, exclusion mechanisms act, and these mechanisms are addressed as Janssen-Cornell effects and are supposed to contribute to the maintenance of tree diversity in species-rich forests and also over long time spans. For BF China, there is evidence that Janssen-Cornell effects act already in this early stage of forest development. So in a reciprocal transplant experiment referring to the monocultures at both sites and thus having sort of home and away situations, we found the performance to vary, like having decreased biomass at home sites while having increased leaf level damage due to herbivory there at the same time. So there is home disadvantage, suggesting strong effects of Janssen-Cornell when tested here on multiple tree species. What we have not yet fully seized is the role of tree species richness for tree recruitment and the associated multi-trophic processes. And this is of importance given that we know that tree species richness suggests sort of bottom-up effects on higher trophic levels, such as arthropod diversity, as was shown already in BF China. For temperate forest study systems, for example, we do know that diversity of tree communities, as shown here for high diversity and low diversity communities, interacts with seedling emergence associated processes, that means with Janssen-Cornell effects. So there's some reason to bring all these different aspects together and to study the role of multi-trophic interactions for tree regeneration, both at different stages of the tree recruitment and along the tree diversity gradient. Our objectives make use of the situation that we do have several reproducing tree species in the experiment available. So in the first objective, we address the demography of early life stages. And here at the observational level, we want to quantify the amount of natural tree regeneration. And we address the mechanisms more in detail at the experimental level by studying the key process of seed predation, both in terms of pre-dispersal seed predation, just as post-dispersal seed predation by means of invertebrate exclosures. And we will finally address also the processes of herbivory experimentally by quantifying Janssen-Cornell effects along the tree diversity gradient.
For addressing these objectives, we have structured our sub-project in 3 work packages. In the first work package, we will follow an observational approach to test the hypothesis that the vulnerability of life stages will vary with tree species identity and change along the gradient of tree diversity. For this work package, we will work in VIP plots and measure sampling density and recruitment success. We will also permanently monitor life stages of key tree species, quantify leaf damage on seedlings and saplings. As core variables, we will measure herb layer productivity and light interception at the plot level. We will also make use of environmental data of the BEF data portal from SP2. In work package 2, we will follow an experimental approach and focus on the key process of seed predation. We pose 3 hypotheses, namely that pre-dispersal and post-dispersal seed predation and the contribution of generalists and specialists to these processes will vary along the gradient of tree diversity. Also, we expect that Janssen-Cornell effects in these processes will depend on community composition of trees and seed predators. For this approach, we will work in the 300 core plots. We will put up seed traps. We will using exclusion bags, namely to collect emerging species from pre-dispersal seeds. And especially with this data, we will establish a bipartite interaction network between fruits and seed predators. We will also be laying out seed cards with and without exclosures, and we will use molecular gut content analysis to also identify the post-dispersal seed predation agents. Work package number 3 will address the key process of seedling herbivory. We hypothesize that with increasing tree diversity, dilution effects next to other frequency-dependent mechanisms will lead to reduced levels of seedling herbivory and mortality. Here we will focus on the 64 VIP plots again and will add seeds and seedlings to all plots with and without exclosures. This way we will create home and away situations with which we will be able to test for Janssen-Cornell effects. Our sub-project is well integrated into the overall project. SP5 provides seed predation and herbivory data to SP3 in order to complement leaf herbivory data, as well as to SP6 in order to better understand the trait-mediated role of ants. SP5 receives data from SP3, namely arthropod seed predators and diversity data, as well as ant abundance data for the tree seed predator network from SP6. We will coordinate our light measurements with SP4 and are well coordinated with Z1 and Z2. SP5 has a Chinese partner project led by Sishu Xiao, and in this project he will focus on vertebrate effects on tree life stages in the canopy and on the ground. He will furthermore address the impact of animal foraging processes on seed germination and survival, and together we will establish the tree life stage interaction web, including vertebrates and invertebrates. We will cooperate in molecular invertebrate gut content analysis, and Sishu Chao has already secured initial funding and will also apply for the joint Sino-German fund.